Welcome to this new Isis Pharma clip. This video will present you a new way to understand and explain homeostasis in its practical application. Now, let's discover how hyperkeratosis and scaly skin can be analyzed with homeostasis and how URELIA acts on the different stages of the pathologies. The keratinocyte renewal starts in the basal layer of the epidermis. It is controlled all along the epidermis by the calcium gradient. The calcium gradient is measured by cell receptors. Its concentration changes with the water content of the epidermis. The drier the skin gets, the higher the calcium concentration. Keratinocytes evolve, mature, and differentiate as they go up the skin layers. In the upper layer, the last stage of differentiation is the granular keratinocytes. Under calcium signal, granular keratinocytes enter into apoptosis and become dead cells called corneocytes. First attached together, corneocytes are peeled off on the top of the stratum corneum. This process takes 28 days for a normal skin. The granular keratinocytes present two types of granules in their cytoplasm. One of them contains a big protein called profilagrin. At this stage, profilagrin is cut into filaggrin, which builds the cytoskeleton of corneocytes. In the stratum corneum, filaggrin is cut into small molecules called natural moisturizing factors. These molecules contribute to making the skin acid. They have the capacity to retain water and are one of the key factors of skin hydration maintenance. The good skin hydration allows a physiological calcium gradient. This balanced system allows the physiological renewal of keratinocytes in 28 days. The origins of psoriasis are not very clear, but it seems that it has genetic origins combined with external aggravating factors such as stress, medication, hormones, or infections. According to the genetic origin, the first step to psoriasis is an acceleration of the keratinocytes turnover. The migration and differentiation takes 5 days instead of 28. This is combined with several abnormal genes coding. One of the consequences is an abnormal filaggrin synthesis. At the last stage of maturation, an inhibition of keratinocytes apoptosis is observed. Keratinocytes accumulate in the stratum corneum, creating the white scaly plaques of psoriasis. Due to a lack of natural moisturizing factors, dryness is observed. Several consequences occur. Abnormal skin pH leads to skin microbiota imbalance and enzymes deregulation. Keratinocytes accumulation induces an inflammation and an activation of the immune system. One of the consequences of this inflammation is a new acceleration in keratinocytes differentiation, turning the homeostasis circle into a vicious circle. The URELIA range from Isis Pharma acts on 3 stages of this vicious circle to help the skin recover its natural healthy condition. Alpha hydroxide acids, also known as AHA, combined to a high concentration in urea from 10 to 50%, offer an intense keratolytic action to remove the keratinocyte accumulation. In addition to this keratolytic action, some AHA and urea are natural moisturizing factors and they restore skin hydration. They are combined with emollient agents and Expert Moist, a specific complex which moisturizes and soothes the skin. At last, urea and the emollient agents have a soothing and antipruritic action that will reduce the inflammation. The specific combination of those active ingredients will reduce the disease's effects on the skin. To conclude, this was our explanation of hyperkeratosis in perspective with the homeostasis mechanisms. URELIA, an effective answer to scaly skin and hyperkeratosis. For more detailed information on the pathology, URELIA, and its mechanisms of action, please refer to the training support available on MyEasy's Pharma. You are now an expert in hyperkeratosis. Please do not forget to consult the other videos and learn more about the other ranges. Easy's Pharma, reimagine your skin.